Welcome to lecture ES-2. In this lecture, we're going to be talking about how hormonal secretion is controlled. We're going to be starting with just a very quick idea of negative feedback and how it is involved in hormone secretion. We'll then use three example systems. The first will be parathyroid hormone, which is a simple negative feedback system. We'll then talk about insulin and glucagon, which are a more complex interactive system. And then we'll go into depth on the hypothalamus and the pituitary and the much more complicated negative feedback systems used by those to control the release of a number of other hormones. Okay, so let's talk about some mechanisms for control of the secretion of hormones. Generally, hormones follow a negative feedback model, where you secrete the hormone in response to some sort of stimulus, and the hormone causes a change which reduces that stimulus, so it brings us close back to some sort of set point. Not always, but often. So we're going to talk about three examples of how that can work. The first one is going to be a very simple negative feedback system. That's how we control the secretion of parathyroid hormone, which is involved in calcium regulation. The second will be a little bit more complex negative feedback, which involves insulin and glucagon, the hormones that are involved in regulating blood sugar. And then finally, we'll talk about the hypothalamus and pituitary gland and how they interact and then interact with other glands in the body in a more complex multi-level negative feedback system. Let's start by talking about parathyroid hormone. Which is usually just abbreviated PTH. So parathyroid hormone is a hormone made by the parathyroid glands, which are a set of small glands on the back of the thyroid. And the main effect of parathyroid hormone is to increase blood calcium levels. It does that by doing three things. One, it increases calcium reabsorption in the distal convoluted tubule in the kidney. So calcium that gets filtered out, we bring more of it back into the blood. It also increases bone breakdown. Parathyroid hormone encourages the cells which break down the mineral matrix of bone and release the calcium into the blood. The third one is that it's sort of an indirect mechanism. It increases the conversion of calcifediol, which is an inactive form of vitamin D, to calcitriol, which is the active form of vitamin D in the kidneys. And calcitriol increases the absorption of calcium from the digestive tract. So this increases digestive absorption. Of calcium. So in all of those cases, the action of parathyroid hormone is to increase the amount of calcium present in the blood. Now, given that, think for a moment about what would it make sense to be the stimulus for the release of calcium, for the secretion of parathyroid hormone? If parathyroid hormone increases blood calcium, when would we want to secrete it? Seems to me that we'd want to secrete it when calcium levels are low. So the negative feedback system would go that we monitor calcium levels. When they get low, we take that as a signal to secrete parathyroid hormone, which brings calcium levels back up. If calcium levels get too high, we stop making as much parathyroid hormone, which lets calcium levels fall. So that's going to be our idea for a negative feedback system for this. Now, how exactly that works, well, that's a different question. We'll get to that in just a moment. All right, so our idea is that this is the basic part of our negative feedback system. The parathyroid gland is going to monitor blood calcium levels, and if calcium levels get low, then we will secrete parathyroid hormone, which will increase blood calcium levels. So the question is, how does that actually work? How do parathyroid gland cells detect blood calcium levels and respond by changing parathyroid hormone secretion? Well, let's answer that. First of all, parathyroid hormone is a protein hormone. So let's imagine that we've got here is a cell in the parathyroid gland. And we will have vesicles with parathyroid hormone in them. So those are vesicles with parathyroid hormone ready to go. We just have to fuse those vesicles with the membrane and release that hormone into the blood. Now another thing in the parathyroid gland is that we will have storage of calcium here inside the cell. And here will be some calcium channels. Now, the way that we're going to do this, we're going to detect blood calcium levels. On the cell surface, we will have a G protein coupled receptor for calcium. This is going to use a phospholipase C mechanism, if you remember how that works. So, blood calcium. Okay, need to switch that one out. Need to switch both of those out. In any case, blood calcium binds to the G protein coupled receptor, which activates phospholipase C, which produces second messengers. Those are DAG and IP3. IP3 binds to the calcium channels, which allows calcium out. Now, normally we think of calcium as turning on machinery in the cell. Like in a synapse, calcium activates the machinery that makes the vesicles fuse and release their neurotransmitter. In these cells, though, oddly, calcium actually inhibits vesicle release. So when calcium binds to the receptor makes IP3, and IP3 releases calcium. That calcium stops the cell from releasing parathyroid hormone, which means that when there is enough blood calcium, we don't release the hormone. So high blood calcium means less parathyroid hormone release. If blood calcium levels fall, that means we don't activate the receptor, which means we don't make IP3, which means I don't release the calcium, which means I don't inhibit the vesicle release, which means I release parathyroid hormone. So when calcium levels are low, that's when I release the parathyroid hormone, which will then, as we talked about, increase blood calcium levels. So there's our negative feedback system. When calcium levels are high, that means we stop releasing the hormone. When calcium levels are low, that allows us to release the hormone. So that helps us keep blood calcium levels at a moderate level.